CH-bin, binning metagenomic sequences using affine and convex hulls. Metagenomics involves the study of various genetic material obtained directly from communities of microorganisms living in natural environments. Metagenomics plays a vital role in applications such as water quality control, clinical disease diagnosis, and pathogen studies in agriculture. The binning step in metagenomics is concerned with separating sequences belonging to different microorganisms into bins such that each bin consists of sequences belonging to one microorganism. Normalized KM count is one of the main features used in binning. Although KMs for values of up to around 6 can retain useful information, existing binning tools rarely utilize K values above 4. The reason for this limitation had been the lack of a reliable and efficient high-dimensional binning algorithm. We tried to overcome this limitation by adapting a novel high-dimensional data clustering algorithm introduced by Sevicalp in 2019. This is the overview of our methodology. We use coverage and composition information of the sequences to determine the bin of a given contig. The workflow of the tool can be broken down into 4 main steps. First, we generate the feature vectors using previously mentioned composition and coverage information. For composition information, we use KM frequencies. For coverage information, we use abundance information. Then, the number of bins are identified. For this, we use single copy marker gene analysis. In the third step, the initial clusters are obtained. This is done via an algorithmic procedure which uses information from the second step. In the fourth and final step, we apply the modified version of Sevicalp's algorithm to perform binning. At a high level, this is how the algorithm works. For each contig, we calculate the convex or affine hull distance from the contig to each bin. We then assign the contig to the bin with the closest distance. This process is repeated for a predetermined number of iterations. In each iteration, we permute the order in which the contigs are selected. This table shows a comparison of F1 scores of other state-of-the-art binning tools against CH-bin. As you can see here, as the general trend, the F1 score for K equals 4, 5 and 6 combination is comparable or better than that of F1 score of K equals 4. It is also worth noting that our binning tool CH-bin in any combination has shown an improvement over the existing state-of-the-art binning tools compared here. We will now look at a demonstration of the GUI tool of CH-bin. You can submit a new binning job by clicking on this button. Then you have to select the corresponding binning tool. Then you have to submit the contig file and the coverage file. Binning will start once you submit those 2 files. You can see the live logs on the right hand side of the screen. After the binning is finished, you can download the results by clicking on the results button. Furthermore, if you want to visualize those results, you can add a ground truth file and then visualize the results. Once the visualization is finished, on the left hand side, you will be able to see the detailed values for the evaluation parameters that are provided by AMBER. On the right hand side, you will be able to see a graphical visualization of the most important parameters out of those. You can also view the results of previously submitted jobs. We hope our video gave you an idea on how we utilized affine and convex hull distances to improve metagenomic binning. Thank you very much.